Good morning. Okay, hope you are all well. Not very good weather outside, but we're here to discuss cell division and hopefully in the comfort of our homes. Now, we've looked at mitosis, which is body cell division, and we should know that when we look at the brief, and I'll put that up now, that we're answering in part B in the test, explain why the process of mitosis is important in cells. And that's the fact that it's for the growth, repair and maintenance of our body cells, and it maintains an exact copy of the DNA and chromosomes for each specialized cell. And the key stages of mitosis are prophase, metaphase, anaphase, and telophase, ending with cytokinesis. Importantly, the two daughter cells that are produced are identical, and they contain 46 chromosomes each. Now, that covers 4.1 and 4.2, and we need to talk about in question 2, part 2, the different stages and the movement of the chromosomes in each of those stages. Now we're moving on to the last question, which is split into 3 questions, 3.1, 2, and 3, and covers assessment criteria 5.1. And first off, it says, name the process in animals which requires mitosis to take place. This is a simple statement. One sentence will do, where we can say the process that in animals that requires meiosis is called sexual reproduction. Now, that also occurs in some plants, but we're not particularly interested in human physiology in plants. So sexual reproduction is that process. Then it says to outline the stages of meiosis and the key chromosomal movements which occurs. But as we'll see, essentially, meiosis is mitosis twice. And in an exam, I wouldn't expect you to tell me again all about the chromosomal movements in mitosis. So we preface our answer with, as meiosis is essentially mitosis twice, the chromosome movement is exactly the same as in mitosis, but there are some essential differences. Okay, and then lastly, we'll explain the biological importance of meiosis. So let's head off to the PowerPoint, which I'll put up on the VLE later today. So let's just get that started. So meiosis is the production of gametes, and gametes are our sex cells. So in male, that's sperm, and in females, that's the ovum or ova. Ovum is singular, ova is plural. And 5.1, the assessment criteria that you'll be marked on, is to relate the key stages of meiosis and its biological importance of biological functions. So let's get into it. Meiosis, the purpose of meiosis is to produce sex cells or gametes used in sexual reproduction. That's our first question. In the male, this is the sperm, and in the female, the ovum. Excuse me. When we look at meiosis, it looks very complicated, but we have a firm understanding of mitosis. It becomes much easier. In mitosis, the cell divides once, going through 4 different stages, as we know: prophase, metaphase, anaphase, and telophase. right, the abbreviation being PMAT. In meiosis, the cell goes through the same stages but twice, and to identify the first and then the second divisions, we use the Roman numerals I and II, which is 1 and 2. So we have prophase 1, metaphase 1, anaphase 1, and telophase 1. Now, those are the same as mitosis, but we'll see that there are some important differences. And then it goes into prophase 2, metaphase 2, anaphase 2, telophase 2. And then we come on to the important differences. So background information. In the early stages of meiosis in the female, so begins when the female is in the mother's womb, the uterus, and the process of developing the female sex actually is called oogenesis. Okay, so the production of the sex cells, the ova, the individual's eggs that a female will produce. Now, when the female, all right, is still a fetus, a baby, all right, they will contain all the ovum, the ovary, in the ovaries. And the process of meiosis 1, however, has stopped at the end of prophase 1. So in a female, that process has already begun, but stopped when they're born. And there'll be no further development until the female goes through puberty, and then each ovum is released in the monthly menstrual cycle. And it will go then through the phases of meiosis 1 and meiosis 2. In the males, however, the process of meiosis 1 and meiosis 2, called spermatogenesis, right, the production of sperm, starts at puberty. There's no pre-stage, all right, for the male. But that's some background knowledge. We've seen in mitosis that there are 4 stages, resulting in the production of 2 daughter cells, each with a full set, complement of 46 chromosomes. This 46 chromosome number is called the diploid number, and you will need to use that specific term. In meiosis, there are 2 divisions. In meiosis 1, we go through the same 4 stages, but in meiosis 2, they're called, in meiosis, they are called prophase 1, metaphase 1, anaphase 1, telophase 1. However, instead of going into interphase, which normally happens in mitosis straight away, the 2 daughter cells immediately start the process again. So they get to the end of telophase, they separate, but instead of then maturing, they divide again. And if you look at the diagram, you can see that prophase 1, metaphase 1, anaphase 1, and telophase 1, and cytokinesis is the same as mitosis. But the 2 daughter cells, as you can see in the second part of the diagram, they go into prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. And instead of 2 cells, we now have 4. Okay, that's an important difference. So the result is that 2 daughter cells divide again to produce 4 daughter cells. However, each daughter cell will only now have half the number of chromosomes, 23, because in mitosis, once the cell's divided, the genetic material, the chromosomes, double up to form the 46. But here it hasn't had a chance to do that, so we end up with 23 chromosomes. Now, this is called the haploid number. Think of H as half the number, haploid. And it's these cells that mature into gametes, or sex cells, we know as sperm and ova. Now, ova is eggs, ovum is one. Now, there is a very important difference that occurs in meiosis 1. And in meiosis 1, during prophase 1, the chromosomes become entangled, they get twisted around each other. And it's called crossing over. And this is where genes can be swapped over. And so sections of genes can be swapped between the identical chromosomes. Now, this crossing over is an exchange of genes, and the genes are mixed up, and it's random, not resulting in a perfect duplicate like in mitosis. Now, this is important in producing variation in the human species and also other complex organisms. It happens in flowering plants, it happens in other animals as well. And it's the reason why we are different from our mother, our father, brothers and sisters, okay? Some of you might think that's a good thing, all right? And it actually is a good thing. Every sex cell made from meiosis has a unique combination of chromosomes. This means that no two sperm or egg cells are genetically identical. Every fertilization event produces new combinations of characters or characteristics. We used to call those traits, which is an old-fashioned name, but you might still find it in some books, but are characteristics, such as eye and hair color and height. Now, this is one of the main drivers of evolution, because if we have all these changes, then if some event occurs which wipes out the vast majority of, say, the human race or an animal species or plant species, there is a chance that some will be genetically able to survive that change, and allows the species to survive. Now, there's one other difference that occurs between the production of sperm and female ovum. In the male, all full sperm daughter cells survive. So every single cell that divides in meiosis 1 and 2 in the male survive. So all four for each cell that it's originated from. In the female, 3 of the daughter cells produced by meiosis are smaller. In fact, what happens when they divide into the 4 daughter cells, 3 of them don't get an equal share of the cytoplasm. So the 4th daughter cell is much larger. Now, the smaller daughter cells are called polar bodies and will not survive, leaving 1 daughter cell that will mature into an ovum each month during that menstrual cycle. So here's a summary diagram, and at the top we've got the labels meiosis 1, meiosis 2. And essentially, remember, meiosis 1, the movement of the chromosomes is exactly the same. So in prophase, we have in both the sperm and the egg, all right? Technically, when it starts, it's called an oocyte and a spermatocyte. The chromosomes appear and the nuclear membrane dissolves. It's there that crossing over starts to take place in both the male and female, and we get that random assortment of genes to give us our variation. So that's a very important difference than mitosis. Then in metaphase, the chromosomes are arranged along the middle of the cell, and we get the actual centrioles holding the chromosomes in the center, the metaphase plate or equator of the cell. In anaphase, the spindle fibers contract and pull the chromosomes to the opposite side of the cell. And then in telophase or telophase, the nuclear membrane surrounds the chromosomes. They start to disappear, okay, and we have two separate cells. Now, that's the same in the female, okay. Now, what then happens is in meiosis 2 is exactly the same, okay. We go from prophase straight into metaphase and anaphase, and we get the dividing of the cells to form 4 spermatids, sperm, okay. They become sperm when they have a tail, the middle piece, the mitochondria, etc. But if we look at the bottom here, only one of the cells matures in the female to form an egg cell, an ovum. Okay, and both the sperm and the egg have half the number of chromosomes, so 23, no doubling up, because when the sperm unites with the egg, we want to restore the 46 chromosomes, so we have a full complement of genetic material. The 3 polar bodies disintegrate and the material is reabsorbed into the body. And the process starts again in the female each month, whereas in male, sperm is continually produced. In the female, only during the menstruation cycle is she fertile, and that's usually from the ages of 13, or that can be earlier in some girls, up to the mid-40s and beyond. With better health care and nutrition, we are seeing women still remaining fertile up into their 50s. So a summary. When we're answering the question, remember, all right, that mitosis is for growth, repair and maintenance of the body, the somatic cells. There are 4 stages, producing 2 identical daughter cells containing 46 chromosomes, called the diploid number. And for 5.1, in meiosis, the essential information is that it's used for sex cells, sexual reproduction, the production of sperm and ova. And there are 2 stages, meiosis 1, where crossing over and random assortment of genes occur, and meiosis 2, where the number of chromosomes is reduced to half, all right, 23, called the haploid number in 4 daughter cells. And lastly, in males, all 4 daughter cells survive and mature into sperm, while in a female, the 3 smaller polar bodies die, leaving 1 ovum to mature and can be released each month. Now that covers 5.1. Okay, now that's going to be on the VLE, and you can go through it. Adrian has also produced, he's another lecturer in the department. He was an advanced skills teacher when I first joined the college. He's come out of retirement. He's actually produced some good animation videos for both mitosis and meiosis. And when you go onto the VLE, remember you can dip into any of the other lecturers' notes. You know, we're not bothered if you can't use other teachers' notes. And on his site, there are videos there that you can look at that he's put onto YouTube. Okay, hope that goes well. I know some people have had problems with uploading the videos. It seems to work better on Microsoft Edge. For some reason, Google still comes up as private and you can't enter it. So look on Microsoft Edge, please. Okay, hope to see you soon. And give me any emails if you are struggling with the assignment. Okay, bye for now.